Hello everyone. In this video, we are going to see about the microbial production of citric acid. First, let's see about the citric acid. Citric acid is a weak organic acid, and the chemical formula of citric acid is C6H8O7. It is naturally occurring component of many fruits and also found in almost all plants and animal tissues. It is widely used as organic acid in the field of food for about 60% and in pharmaceutical for about 10%. W. Shields in 1784 first time isolated citric acid from lemon juice as calcium citrate. It is the molecular structure of citric acid. In biochemistry, it is an intermediary in the citric acid cycle that occurs in metabolism reactions of all types of aerobic organisms. It is a type of organic acid which accumulates during the controlled fermentative growth of particular species of Penicillium and Aspergillus. Let's see the properties of citric acid. The molecular formula will be C6H8O7 and the molecular name is 2-hydroxy-1,2,3-propane tricarboxylic acid and the molecular weight is about 210.14 gram per mole. Occurrence, it occurs as a metabolite in certain plants and animals. The melting point will be 153 degrees Celsius and it is soluble in polar solvents like water when citric acid is in pure form. The chemical state, it will be a solid state at room temperature. The production either by chemical process or by microbial process through the fermentation. Citric acid production. Production of citric acid is the process of extracting or manufacturing citric acid via natural and synthetic method or chemical method. The natural process is a technique through which we can extract citric acid naturally from the citrus plants like lemon, orange, etc. And in synthetic process is an alternative technique through which one can produce citric acid chemically via enzymatic synthesis and biologically through the fermentation by the microorganisms. Now let's see about the production process of citric acid. There are two types of production process, a biochemical method by fermentation, a biological by chemical reaction. Biochemical method. In a biochemical method, citric acid is produced as a primary metabolite by the microorganisms. The microorganism which produce citric acid during trophophase cell growth because of defective citric acid or Krebs cycle. In defective Krebs cycle, a high amount of sugar is transported through the BMP pathway that form acetyl-CoA. The formed acetyl-CoA condenses with oxaloacetic acid to yield citric acid by the activity of an enzyme citrate synthetase. It is the biochemical pathway, glucose which produce pyruvic acid by the means of EMP pathway, then acetyl-CoA plus oxaloacetate is formed by the condensing enzyme, then citric acid is formed. Biological method by the use of our microorganisms. The microorganism used include yeast, that is Candida species, and bacteria like Bacillus licheniformis, Arthrobacter paraphenans, Corynebacterium species, and fungi like Aspergillus niger, Aspergillus aculatus, Aspergillus avamori, Aspergillus carbonarius. The medium used for the fermentation include it requirement of high production, that is carbon source like beet molasses or sugar solutions. Na-ferrocyanide is added to reduce ion 1.3 ppm and manganese less than 0.1 ppm. It need highly dissolved oxygen concentration and high sugar concentration. The lower the pH value at the time of production phase, that is pH less than 2, which minimize the risk of spoliation by different organisms and inhibit the manufacturing of unwanted organic acids. It also needs the temperature for about 30 degrees Celsius. Nitrogen and phosphate limitations, like few complex media, are rich and rarely need to be supplemented with a nitrogen. Trace elements like zinc, magnesium or manganese, ferrous ion, that is copper, and heavy metals. Bioreactors use may be batch or fed batch. Aeration is provided to the fermenters by air sparging. Agitation is to avoid shear damage on molds. Citric acid production techniques, that is surface fermentation, submerged fermentation or solid stage fermentation, can be used for the citric acid production. In surface fermentation, it is also known as surface culture process. It is also called liquid surface fermentation. Surface culture fermentation was first mentioned introduced for citric acid production in 1919. In liquid surface fermentation, the culture medium, that is pH is about 5 to 6, is added to the aluminum shallow trays up to 5 to 20 cm deep. The process is carried out in the fermentation chamber, which provide uniform air circulation, maintains relative temperature and humidity. First, the spores of Aspergillus niger is blown onto the surface of the culture medium for about 5 to 6 days, and dry air is then passed. Then the pH of the culture medium is adjusted between 1.5 to 2 pH. After 24 hours, the spores start to germinate and the growth of white mycelium can be seen on the culture medium surface. After the utilization of sugar content by the moles, the remaining liquid is separated from the mycelial mat. In the surface culture process, a small amount of citric acid is produced as a primary metabolite by the Aspergillus niger. Then in solid state fermentation, the solid substrate is soaked with water up to 65 to 70% of water content. After the removal of excess water, the mass undergoes a steaming process. Sterile starch paste is inoculated by spreading Aspergillus niger conidia in the form of aerosols or also a liquid conidia suspension on the substrate surface. The pH of the substrate is about 5 to 5.5 and the incubation temperature is about 28 to 30 degrees Celsius. Growth can be accelerated by adding alpha amylase. Although the fungus can hydrolyze starch with its own alpha amylase during the citric acid production, pH dropped to value below 2. The solid state surface process takes 5 to 8 days, at the end of which the entire is extracted with hot water. On other cases, mechanical passes are also used to obtain more citric acid from the cells. Then we see about the submerged fermentation. Beet molasses substrate, that is 12 to 15% reduced in sugar content. Nutritive salts such as ammonium nitrate or potassium dihydrogen phosphate are added. The pH of the substrate is maintained at 5.5 to 5.9. The process can usually run in one or two stages using hydrophilic spore suspensions or germinated conidia from the probacata stage. Amount of spores are 5 to 25 into 10 to the power 6 per liter of substrate. The development of the hyphae and the aggregation generally requires a period from 9 to 24 hours at temperature of 32 degrees Celsius. Then mycelial aggregation and spherical pellets, their productive form can be detected after 24 hours of inoculation. The change of pH in this phase is from 5.5 to 3.5 for beet molasses substrate and to 2.2 from the sucrose substrate. Fermentation lasts up to 6 to 8 days and later citric acid is purified from mycelium. It is the pathway. First, stock culture of Aspergillus niger is needed. Then, culture sporulation on PDS land, that is potato dextrose agar land. Then, spore inoculations, that is in 0.9% NaCl. Then, it is subjected to fermentation. It contains 75 ml beet molasses in 250 ml buffer shake flask at 30 degree for 8 days. Then, substrate beet molasses is used. Then, after a mycelium is formed, it is undergone to filtration and the supernatant citric acid and sugar will be the supernatant and the residue fungal biomass, it will be act as a animal feed. Recovery. First, it involves the removal of fungal mycelium from the culture medium, then filtered to remove mycelia and precipitated as oxalate. Resulting solution is heated and CaO is added to form precipitate of calcium nitrate. Filtration and treated with H2SO4 to generate citric acid and precipitate of CaSO4. Filtration of dilute citric acid solution is decolorized and then it is evaporated to produce crystals of citric acid. Crystals recovered by centrifugation are then dried and packed. Then uses of citric acid. It is used in beverage industries to provide tartness and complements of fruits and also increase the effectiveness of antimicrobial preservatives. In jellies, jam, and candy, it provides tartness to the products, involves in pH adjustment and minimize sugar inversion. And in frozen fruits, this acid lowers the pH to activate oxidative enzymes and protects ascorbic acid by inactivating trace metals that is found. In dairy products, it is used as an emulsifier in ice cream and processed cheese. In pharmaceuticals, as effervescent in powders and tablets, and also used as anticoagulant agent. In cosmetics and toiletries, in pH adjustment and buffering agents. Thank you for listening.